with that, we'll move to our next presentation. Sean and Varsha from Johns Hopkins will be presenting next. So let me just get our settings ready, and then I'll hand the podium over to both of you. Go ahead. Thank you.
Hi, so today Varsha and I will be presenting on developing a multi-stage mRNA vaccine targeting the AMA1-RON2 complex and CSP. And AMA1 is critical for both sporozoite and merozoite invasion during this process. RON2, as shown, you get some mouse here. Oh, there we go. As shown in light green, is inserted into the host plasma membrane. And AMA1, shown in dark green, binds to a 47 amino acid region of RON2, which regulates parasite by initiating tight junction formation. A previous phase 2 clinical trial of an AMA1 vaccine demonstrates that vaccination with E. coli expressed AMA1 produces robust antibody titers, and AMA1 vaccination shows modest but insignificant decreases in episodes of clinical malaria against malaria strains similar to 3D7 used in vaccination. There are two problems to overcome for effective AMA1 vaccine. The first being developing a vaccine that can target many different types of strains. Additionally, the phase 2 clinical trial against AMA1 shows only modest and insignificant efficacy against clinical malaria. I'm just going to skip these slides for time. We'll take a quick look at here on the right. This is looking at a falciparum challenge of Aeotis monkeys challenged with AMA1 in blue as compared to the AMA1-RON2L complex in red. And you see a significantly delayed patency or development of parasitemia in the AMA1-RON2L complex group as compared to the AMA1. Skip that slide for time. AMA1 consists of 3 domains. In the bottom, we see domain 3. In the top, we have domains 1 and 2, which surround RON2L in orange. Domains 1 and 2 are known to interact with RON2L. We tested if the increase in neutralizing ability that we're seeing with the addition of RON2L only requires domains 1 and 2, or if it requires domain 3 as well. Interestingly, we see a increase with only domains 1 and 2. Additionally, we find when we vaccinate mice with domains 1 and 2, a significant decrease in the development of parasitemia in P. Yolii challenged mice, shown in the bottom right there. We then designed AMA1-RON2L fusion proteins. On the left, we have fusion protein 1 with RON2L inserted between domains 1 and 2. On the below it, we have fusion protein 2 with RON2L inserted in the opposite orientation. We did an ELISA shown in the middle, looking at the ability of free RON2L to bind to fusion protein 1, AMA1, and fusion protein 2. As expected, we see significantly decreased binding to fusion protein 1 in the middle, and moderate decreases in binding to fusion protein 2. And when we do X-ray crystallography of our fusion protein 1 and fusion protein 2, done with the gracious help of the Bloomberg lab at the University of Victoria, we see that the structure of fusion protein 1 forms crystals with RON2 mirroring almost identically, besides a few angstrom differences at 2 specific points. The structure of the AMA1-RON2L complex, however, fusion protein 2 did not form any crystals, suggesting a more loosely held structure. Interestingly, both fusion proteins induced high levels of antibody titers on the left shown there, and also high levels of neutralizing antibodies. And here we look at concentrations of 5, 2.5, and 0.83 milligrams per ml, and we see high levels of neutralization for both proteins at all points. Additionally, we see that antibodies to the fusion protein target more conserved regions of AMA1. On the left, this is data from antibodies purified from rats vaccinated with either domains 1 and 2 or our fusion protein 1. And we're looking at the ratio of heterologous to homologous binding. And we see that in fusion protein 2, we have a significantly higher level of heterologous binding than domains 1 and 2. And if we look in the top right, we also have the 1E loop, which is known to be a highly conserved domain of AMA1. And we see for both fusion protein 2 and fusion protein 1, significantly greater binding to the 1E loop in the fusion protein versus AMA1. And we then wanted to look, we know we have neutralizing antibodies. How do they compare to AMA1? AMA1 is shown in blue in the bottom left. Then we have the AMA1 RON2L complex in green, followed by the two fusion proteins. Surprisingly, and we didn't expect this, we see greater neutralizing ability for antibodies against AMA1 than both fusion proteins or the previously studied AMA1 RON2L complex. However, we have strong variation, you can see, in our neutralizing ability per rat within the fusion groups. Why could this be? We didn't expect that result, as we mentioned. Our previous studies used Freund's adjuvant. Our current study is using Adavax, much less immunogenic adjuvant. And when you plot out the different rats, you surprisingly see that the overall immunogenicity of the vaccine appears, it should say, directly proportional to the IC50 of the fusion samples shown in red, but not AMA1 shown in blue. We then reasoned that we must, in order to replicate those early results we saw in the Freund study, develop a platform that also uses a very highly immunogenic platform, platform. And so to do this, we created a mRNA vaccine targeting the AMA1 RON2L fusion protein. We created two different types, a membrane-bound and secretory form. And IFA data shows circumferential expression of the transmembrane fusion protein in HEC293 cells, and more diffuse, as expected, expression of the secretory construct. And when we, following immunization of mice with these vaccines, we found that membrane anchoring the antigen may improve the immune response. When you look at bleeds 1, 2, and 3, which were done at 2-week intervals following each immunization, you see a significant increase in the transmembrane groups as compared to the secretory groups for all 3 bleeds. We interestingly did not see any difference between IM and sub-Q immunizations. And I'll just touch on briefly on the right. We found as a side result that with double the dose of liposomes, we also get a significant increase in the immunogenicity of the vaccine. And similar to the fusion protein, we perhaps see more antibodies to conserved epitopes when looking at a heterologous vaccine, at a heterologous antigen like FVO over 3D7. And I will hand it off to my friend Varsha for the next part.
So as mentioned by Sean, in our aim to create a multi-stage vaccine along with the AMA1 based construct, we selected CSP, the circumsporozoid protein, which is a part of the only approved RTSS vaccine and has shown efficacy against the liver stage of the infection. 2 CSP mRNA constructs provided by Dr. Markham's lab was utilized for the study. One of these CSP constructs contained a MIP3 alpha tag. The MIP3 alpha is a chemokine that rapidly interacts with the CCR6 receptors on immature dendritic cells. Previous studies from Dr. Markham's lab have shown improved immunogenicity of the MIP3 alpha containing constructs, and hence we hypothesize that this tag would increase the uptake by immature dendritic cells and enhance the immunogenicity. So to evaluate these constructs, we performed an in vitro transfection of HEX cells, and both the constructs show expression as seen here. Furthermore, groups of mice were vaccinated thrice at an interval of 2 weeks, and 2 weeks after the final vaccination, ELISA was performed against full-length CSP protein and the NA-NP repeat peptide. So as seen here, the CSP, So as seen here, the CSP MIP3 alpha appears to have more antibodies towards the NA-NP repeat peptide in comparison to the CSP mRNA. Alongside this, we also performed a luciferase assay with P. Berghei parasites expressing P. falciparum CSP provided by Dr. Zavala's lab, and quantified the rate of liver infection 40 to 42 hours post-sporozoite inoculation. So as seen here, the mice vaccinated with CSP MIP3 alpha show lower liver burden in comparison to the mice vaccinated with the CSP mRNA. The same data shown in percentage of inhibition shows about 70% inhibition, sporozoite inhibition in CSP MIP3 alpha group compared to about 40% in the CSP mRNA vaccinated group. So this indicates that the immature DC targeting CSP shows greater inhibition of the liver stage infection. So with this promising data, in future, combining AMA1 based mRNA vaccine with CSP based constructs could prove to be more effective vaccine strategy. So with this, this is our future direction. So we plan to finish our GIAs with the mRNA constructs, and as well as we have a new construct of self-amplifying RNA, and combining this with fusion protein could also be a good vaccine strategy. And all these vaccinations were done with LNP, hence we are planning to actually decide the dose of LNP needed for optimum immunogenicity. And as mentioned, we are planning to combine the mRNA expressing AMA1 fusion protein with mRNA expressing the MIP3 tagged CSP as a strategy for multi-stage vaccine. With this, I would really like to thank Dr. Weizmann's lab, Dr. Buller's lab, and Dr. Richard Markham's lab for their immense contribution towards this work. They provided us with the constructs, and as well as Dr. Godfrey, who helped us with the mosquito work. And along with this, I would like to also thank our current lab members and past lab members for their continuous support. Thank you so much.
Thank you both so much. I think we have time for about one question or two if there are any, room or online. Yes, Jeff.
Yeah, thank you very much. This is really exciting work. I'm not from this field, but I'm very interested in vaccines and vaccine deployment. And everybody knows the RTSS vaccine has less than ideal efficacy. And what has been shown is that the greater antigenic variation between like the strain that was used to develop the vaccine versus what is circulating has big impacts on the vaccine efficacy in the real world. So I'm just wondering if those types of considerations are being put into this type of work. And in relation to that, I hear this a lot, like it's a highly conserved region, like you had mentioned it in like the first part of your talk. And I'm wondering if there's a way to quantify how conserved that is, because it kind of sounds when people talk about highly conserved that it's a dichotomous, it's either conserved or not, but it's obviously a very big gradation in there. So when you say highly conserved, how conserved are you saying those regions are that you're targeting?
To clarify, with respect to CSP or AMA1?
I guess to both, but you had mentioned it in your AMA talk, so you could talk about that.
Okay. So AMA1 is actually one of the most, probably the most polymorphic protein within the Apicomplexa species. 4 of the 5 most polymorphic. It's conserved in all Apicomplexa species, but 4 of the 5 most polymorphic regions are actually in AMA1. best I can say is that it's highly polymorphic, and I think we may be out of time. I'm getting a signal.
Thank you so much.